Okay, we're recording. So we are continuing our survey into NGS data. And today we're going to talk about finding biological meaning from RNA-seq. And so we're going to do some differential expression analysis. We're going to see here how to import your counts and manipulate those tables into a form that we need to do differential expression. We're going to describe the tests and why we do certain things to do this differential expression statistical test. We're going to look at our different methods of visualization and understand how we can find biological meaning from those plots, from the visuals. And then we're going to talk about functional enrichment analysis. Can we take what we found in differential expression and ascribe some biological meaning to that? Okay. So we're doing things a little bit backwards here. We're starting with some publicly available data that's already been analyzed down to the point of counts. And so when we talk about counts, we are talking about counting those individual reads mapping to gene regions. And for most of these reads, they'll map to a gene region. They're going to be easy to count. In these examples right here, all of these red short reads, some of them are aligning to gene A, the chromosomal region of gene A. Some of them are aligning to the chromosomal region of gene B. And those are easy for the computer to count because they are clearly one gene or another. There are some other instances where these aligned reads are harder to assign to one gene or another. And that is because the structure of the genome is complicated. If we remember that the DNA gets chopped up in different ways to make RNA, and the RNA is made up of exons, and it could be made up of different combinations of exons, and genes can overlap, and genes can, one gene could be going this way and the other gene could be going the other way. So the gene structure is complicated, and we could have a read that mapped to maybe where two genes are overlapping. And in this case, it's ambiguous to the computer and to us whether this read right here belongs to gene A and gene B. So we can't count it for either one. There also could be an instance where a read, we're trying to find a place for that it exists in the genome. We're mapping it to the genome. It could be that this read maps pretty well to this location on gene A, and it maps pretty well to this location in gene B. And this is an instance of multi-mapping. One read mapping to multiple places. Now, when I do these countings, I usually tell the program, if a read is multi-mapped, mapped to different locations in the genome, assign it to the one place where it has the highest quality of mapping. And this is called the primary alignment. And that's what I ask the program that does the counting to use. That way I can rescue some of these reads. And even though it's covered up here by this hide meeting controls, even though we assume that we are sequencing the RNA in a sample, and this would be only the official gene regions, there could be times when a read maps completely outside of gene regions. And this could be a bad alignment. It could be a false alignment. It could also be some gene that we don't know about yet. Maybe a part of a gene, a brand new exon that scientists have never discovered exists right here, and this read maps to it. but in our genome annotation, it doesn't know it's there. So this gets classified as a no feature. We can't use these reads. So we're counting reads mapping to known gene locations. And that is what we're downloading from these tables from publicly available data. And we're going to use differential expression. Now, later on in this class, we're going to take a step backwards and we're going to say, how did we get these counts? We're going to have raw reads, we're going to map them to the genome, and then we're going to count them. But we're not to the point where we're going to learn about that kind of computing environment. So we're just being really simple here. We're assuming that you download publicly available data. You just want to do some differential expression analysis. And this will be useful when you're thinking about your final projects, because I don't expect you to download raw data and do all of that process. Now you could, but that would be really hard for the limited amount of time that we have for this class. So to make it easier for your final projects, I suggest you find some publicly available NGS data and do some kind of analysis with it, rather than going from that super raw data and doing computationally heavy mapping. So that's why we're starting out learning about what can you do with counts. Okay, so for this week's lecture for the hands-on R differential expression activity, I downloaded TCGA data from the Xena data portal. You can download it directly from there in your browser, doing the right click and download. You could also, if you have access to a computer, the supercomputer environment, which we'll get later this in the class, you could use wget and download directly into your Linux environment. That's what I did because it's easier. And then we download that data. We have to process it a little bit. And I always say getting the data tables into the processed, into the setup, usable by the differential expression programs is actually the hardest part. But we have to take these sample names, we have to cut them up, we pull out certain parts. And so we have some pre-processing to do. And I'll go through that when I'm going through the script in the next lecture. But there are a few steps that we have to do to pre-process this data. We read in the counts, we do a little bit of math, we do some chopping up of sample names, and we do some filtering. So we want the tumor samples and the normal samples from our table. And we know this because it's part of TCGA's guidelines that all tumor samples are going to have 0, 1 as part of the name, and all normal samples are going to have 11 as part of the name. So that's what we do at the beginning, just to process our data. We have to, so something that's not entirely obvious on the Xena data portal is the numbers they provide you with are not raw counts. They provide you with raw count +1 and then log 2. And for differential expression, using this DEC2 package, we need raw counts. And so I unlog this value, I subtract 1, and then I round it to 0 because this needs to be a whole number to be a count. And DEC2 will complain if it's not a whole number and not a count, because the assumptions that DEC2 makes is that the input is raw counts. So there's that. The program we're going to be using is a package from Bioconductor. It does differential expression analysis, and it assumes that most genes are not contributing to the difference between groups. And I'm going to walk you through this analysis. There's also very nice vignettes and tutorials on how to use DESeq2 in more complicated setups than I'm going to be presenting in this class. So those options are there for you to look at. This is the form, the commands that you would use to run DESeq2 differential expression analysis. So the things you're going to need a counts table. We have that right here. You're going to need a sample table here, and you're going to need information on what are the groups that each sample belongs to, and that is bound up in this sample underscore type dot samples design matrix. And we put all of those things together. We use this DESeq data set from matrix function, and that's going to package everything up into a DESeq2 object. And it's a combination of data objects. I think it's a list. So it's all packaged up, and now we can do DESeq2 functions, apply those to this object. First, we estimate the size factors. So this is going to correct for any sample to sample library differences. So I'll show more about this on the next slide. No, in a couple of slides here. Okay, so we'll get to that in a little bit, talking about size factors. The second thing we're going to do is estimate the dispersions. So we're going to look at the biological variance measured across for each of these genes. We're going to look at how does this vary. And since DESeq2 assumes that most genes are not going to be changing between the groups, it's going to apply a shrinkage to these measures of variance and shrink them to be closer to the average. Then it's going to fit a negative binomial general linear model and apply walled statistics. This is doing the actual statistical test to check every single gene in our data table to see whether that gene is different between our two groups. And then after doing that, we will extract the results from this object, and it will give us the measurements for every single gene, telling us what is the fold change between groups, what are the p-values of that statistical test comparing these two groups. So in the example, I go through these steps one at a time. But if you're doing this in practice and you just want to simplify things, you can replace these three commands with one DESeq function, and that will run through three commands all in one. But you still have to pull out the results from that object. So once you have results, you can ask R, give me a summary. I want to have a p-value cutoff of 0.05 and tell me how many genes did I have that met that cutoff level. This is the output of that summary. Since we're using TCGA data, we have a very large sample size. We have groups that are very different, and we get a lot of genes that are differentially expressed at this adjusted p-value less than 0.05. 41%, they have a positive fold change and meet that 0.05 cutoff level, and 25% have a negative fold change and meet that adjusted p-value cutoff level. So we're getting a lot of genes differentially expressed in this data set. So these are the things, I've already said this, the things we need for differential expression. We need sample groups with replicates, at least 3 per group. That's the absolute minimum that I would even consider doing differential expression for. You need a counts table, so these need to be whole count, not normalized counts yet. Now, if you're downloading data from something like Geo, back several years ago, Geo wanted us to upload normalized expression tables. And if you want to do your own differential expression analysis, that makes it very hard. But I've noticed recently more and more researchers, when they upload to Geo, they upload the counts table. And that's so nice because you can download that counts table. You can do your own differential expression analysis. Without a raw counts table, you would have to download the raw FASTQ files, those raw reads, do your own alignment, do your own counts generation, just so you could do differential expression with somebody else's data. So counts are what you need. The third thing you need for differential expression analysis is that sample information. You need groupings for your samples. So every single sample, you need to know whether it belongs to group 1 or group 2. And your group 1 and 2 will be defined by your experimental question. So in our case, it's tumor and it's normal tissue. And so when you're putting this data into DE-seq2, it requires that the number of rows of samples equals the number of columns in your counts table. Because the counts table is set up with rows as genes and columns as samples, where the sample table is set up with rows as samples. And so they need to have exactly the same number of samples, exactly the same samples, and they need to be in the same order because DE-seq is assuming that the rows of your samples equal the columns of your counts. And so I do a silly little merging and splitting to make sure we have the same sample set and they're in the same order. And then you need a design. And generally, your design is pretty simple. There's 2 groups. Sometimes you'll want to add in covariates. So you're testing your tumor normal groups, and you also want to adjust for sex or age, or the knowledge that these tumors and normals come from the same sample base. And that's what we have here. We're telling it correct on the knowledge that the tumor and the normal come from the same person. You can have a simple design, or you can look into having a more complex design. Okay, so we are talking about counts aligning to genes. And so we have 1 count for every gene in every sample. And it could happen that in 1 sample, you have a very low number of counts. And in another sample, you have a very high number of counts. And this is just because some technical issue on the sequencing machine. So there are differences in total number of counts for each sample. We have a way of handling this because a low number of counts just overall in 1 sample and a high number of counts just overall in another sample is going to follow through onto our gene-wise counts. So gene A in our low sample is going to have a low count, where gene A in our high sample is going to have a high count, even if these 2 samples had the same amount of gene A. So we need to normalize out those sample-to-sample differences. And that's what we're showing here with 1 and 2 in this figure. Since we are only comparing 1 gene in our 2 groups, that's all we have to care about. We only normalize the sample-to-sample differences. If you wanted to compare gene A to gene B, then you would have to be concerned about what is the length of these genes. So a short gene, just because it's short, will have fewer reads, and a long gene, because it's long, will have more reads. And if we want to compare a short gene to a long gene, then we have to do some gene to gene normalization. And that's what we have when you might hear about FPKM, fragments per kilobase of gene length per million reads per sample. So that's normalizing for the length of the gene and the depth of the sample. You also might hear about transcripts per million, and that also normalizes length of gene and coverage of sample. But in our case, we are only caring about coverage of sample, and that's what DEC2 normalizes for. These are the steps that DEC2 takes to normalize out those sample-to-sample differences. It looks at all of your samples and makes the geometric mean of all of your samples, and that is what we call the pseudo sample. And then it compares every single sample to our pseudo sample, making a ratio, and it calculates a scaling factor based on that ratio. And then it divides the count of each gene in a sample by the scaling factor of that sample. So that's how it's normalizing out the sample-to-sample differences. If you get a table of data and somebody says, hey, this is my counts table, a quick check is this table, does it have whole numbers? If it has whole numbers, you're probably pretty safe in assuming this is counts. If the table has decimal numbers, then I would look into it further because there is something not whole about these values. They're either normalized already, or maybe they've been log transformed like our Xena data that we have to deal with a little bit later. So if you see decimals, question it. Is this really counts? Do I need to do something to this data set to bring it back to counts? So in our script, we'll go over later, we import the data into our DEC2 object, we normalize it, we process it. The first thing we're going to do is we're going to apply an additional stabilizing transformation just to get the expression values out so we can look at the principal components plot. This is a method of taking all of the dimensions of all of our genes and squishing it down to a two-dimensional view. And from this view, we are asking, how do our samples cluster together? And this collapsing onto a two-dimensional view makes a measurement for every single sample of the first dimension, the variability in our data set. This is PCA1 or PC1, and then also the second most contribution of variation in our data. This is called PC2. You can actually calculate out a whole lot of PCs, like 3, 4, 5, and whatever, but usually we just look at PC1 and PC2 and ask, do these two main lines of variation in our data, are they explained by our known sample grouping? So when you make this PCA plot, you can shade it based on whatever clinical factors you want. So the first thing I'm sure you would want to do is shade it based on our groupings that we're doing differential expression on. And here in this case, I'm shading them based on primary tumor in blue and normal tissue in salmon. And in this example, I notice there is a blue sample just hanging out with our normal tissues. And so this seems like a problem for us. It's either a sample that was mislabeled, or it's a tumor sample that when it was dissected out from the tissue, it held on to a big chunk of normal, and that just got processed in. So now this particular tumor looks a lot like a normal sample. So you're doing this analysis, you need to make a decision. Do you leave the data as it is, just knowing that this one tumor is over here, it might affect our results a little bit? Or do we take this tumor out? And in this example, where it's split so well here, and this tumor is really embedded into this normal cluster, I would just remove it. Since we have a big enough sample size, removing one sample is not going to impact this analysis very much. So in this case, I would remove the sample. If you had a data set that had 3 tumor samples and 3 normal samples, and one of your tumor samples was maybe out here, then in that case, I would not remove it because you already have a low sample size and it's only slightly over that way. So this PCA plot is a good way to get an idea of how do our samples look like other samples in the data set, and is there any suggestion that we might want to trim our sample set a little bit if there are any glaringly obvious outliers like this. Then something else I observe from this plot is in our tumor samples, there's a lot of variation. They're diffuse, they're a little bit farther away from each other. Where our normal samples, they are tightly clustered over here. So normals are really very much like other normals, where tumors, they're more like other tumors than any other normals, but there's a large amount of variation. Like the James Cancer Center, their catchphrase is there's no routine cancer. And we see that right here. Every cancer is a little bit unique. And we are trying to find in this analysis what makes all the cancers similar and different from the normal samples. Those are my observations from this PCA plot. So this is getting to that point of what do we want to accomplish? So the goal of differential expression analysis is to test, to determine for each gene, is the difference in expression between our 2 main groups? Is there more difference between these 2 groups than the amount of variation we observe within our groups? And we have to realize that there is variation in our data. There is biological variation. So one person is not exactly like another person, and there's going to be person-to-person differences that just are because they are different people. That biological variation will get smaller if you use something like a mouse or a zebrafish, because they are bred to be more like each other. So there's biological variation. There's also technical variation. So there's differences in sequencing from day to day, from batch to batch. So we have to think about that technical variation and the biological variation. So we need replicates. Biological replicates are always needed. There is a lot of uninteresting variation in the data. So we need enough consistent measures so that we can understand, is there something here making interesting variation? Okay, so just a little bit about statistics. I know I don't like statistics. I'm sure lots of other people do not like statistics. So we are trying to find a difference between the 2 groups. And to do this, we take what we assume to be a good distribution for the data and try to apply that to our data and ask, is there a difference between those groups? And so let's think about some of the distributions that exist in statistics. So there's a binomial distribution. It's used a lot with counts data. Like if you are flipping a coin, how many times are you going to get a head compared to a tail? But that doesn't really apply here. There's reasons. I will tell you reasons later. So there's also the Poisson distribution. It's used in counting situations as well, but when the number of events is very large, and the probability of having the event is very small. So think about the lottery. You could buy a lottery ticket, but you're probably not going to win because out of the millions of people who buy the ticket, only one person wins. So in our RNA-seq data, we do measure a large number of things, a large number of genes, and the probability that any one of our gene is causing this disease is very small. So we think maybe the Poisson might be appropriate, but there's a catch. The Poisson assumes that the average in your data is related to variance directly. It's a 1-to-1 ratio. As you get a higher, but is it the Poisson distribution assumes that there is no, that the mean equals a variance. But we see with RNA-seq data that genes with high expression will have a higher variation than genes with lower variation. So there's a relationship between the expression and the variation. So the people who decide statistics have decided that a negative binomial distribution is the best model for our RNA-seq data because it allows that the variance can change with the mean. Or if we were going to use the binomial distribution, that would require that the variance is less than the mean. The Poisson assumes that they are equal, and the negative binomial distribution allows that the variance can be greater than the mean. And if we looked at an example of in RNA-seq data, how does the variance relate to the mean? So we have the scatter plot with the mean on the x-axis and the variance on the y-axis. If mean was equal to variance, it would follow this red line here. I think this is a 1-to-1 line. It doesn't do that. The variance is clearly higher than the mean. I think this is made up data, but if we were to look at our own RNA-seq data, it would probably look like this too. So know that the statistical test that DE-seq2 is doing is the negative binomial, since in RNA-seq data, the variance is often higher than the mean. That's why we do it. And also, when we do a statistical test, you'll always see it, we get a p-value. And this p-value is the probability of what we see, is the probability of getting results that we see. So this is fine if you're testing one gene, but we are testing maybe 30,000 genes. And so probability of seeing this happen once is fine, but if we're testing things over and over again, we need to account for all of these different tests that we do. And so we apply a multiple testing correction to make sure that the change we see is consistent enough in our data set to be a true difference. And what we use most often is this FDR, Benjamini-Hochberg correction. And so what it does, it ranks all your p-values and it multiplies each of your measured p-values by the total number of genes divided by the rank of that p-value. And it adjusts your p-value to something a little bit higher. And so using adjusted p-value is the safest option for filtering your data set. Okay, so we have put all of our data into a DE-seq2 object. We've looked at the samples in our PCA plot. We've done our differential expression statistical test, and that provides us with a p-value, an adjusted p-value, and a fold change. So one of the ways that we like to view our analysis is with a volcano plot. In the volcano plot, we'll plot the fold change on the x-axis. So a A higher positive fold change will be on this end, and a more negative fold change will be on this end. And then it plots the p-value on the y-axis. But this is the negative log of the p-value. And we use adjusted p-value here because that is the better option. So we are taking the adjusted p-value and we are applying a log 10 transform of it. And what this does, since it's the negative log 10 of our very small number, the smaller our p-value, the larger the negative log 10 of that p-value. And so our most interesting genes are going to be in these upper left and right quadrants of the volcano plot. Over here will be our most differentially expressed positively changing genes, and over on the other side will be our most differentially expressed negatively changing genes. And the direction of positive and negative is determined by your grouping information. So you should set your group as a factor, and you should set the levels of your factors. So you have control over which group is positive and which group is represented by the negative. If you don't set the levels, it's just going to be alphabetical, and it won't be obvious until you're trying to figure out what in the world a positive fold change means in this data set. So I've done that plenty of times, done my analysis, and then gotten to the end, and I'm like, I don't know which group is which. So there are some ways we can check that at the end. But if you just set it from the beginning, it's going to make your interpretation a lot easier. Okay. So this is representing all of the genes that we tested in our differential expression analysis in a volcano plot. Every dot is a gene, and its location is determined by that one gene's fold change and p-value. Then our next visualization option is going to be in a heat map. And for a heat map, we want to make a selection of genes that we think are the best options for our differential expression subset of genes. I suggest, at a minimum, having adjusted p-value of 0.05. Check that, see how many genes you get with that. If you get more than would fit on a heat map, maybe you need to be more strict. Maybe you need to add in a fold change cutoff. Maybe you need to make your FDR adjusted p-value cutoff smaller. In this case, we're comparing those tumor versus normals, which are very different, and I get an extreme cutoff here. I filter the log 2 fold change. I want it to be greater than 3 or less than -3. And I cut off the FDR, in this case, 1 times 10 to the -50. So that's ridiculous cutoff. But to get a reasonable number of genes, I had to be ridiculous here. The program that I'm using to make heat maps, it's going to complain if you have more than 2,000 genes. So I had to do these cutoffs to get a reasonably low number of genes for our heat map. But for a heat map, you need a subset of your genes that you think are the best representation of genes that are differentially expressed in your data set. We get, for those genes, we get the normalized expression values, and we scale the normalized expression values so that for each gene, each row here is a gene, it's going to find the average of that gene and shift the average to 0. So for this one row, it's been scaled so that the average of this gene row is 0. If a sample has an expression of that gene that's above average, it's going to be shaded in red. So these are all expressing above average. If a sample for that gene has an expression below average, it's going to be shaded in blue. So all of these genes, they're below average for these green samples, and they're above average for the gray samples. And with this function to make a heat map, I've set up some heat map annotation, and that generates this green and gray track. I've told it, so each column here is a sample, and I've told it if the sample of this column is a tumor, make our annotation track green. If the sample of this column is a normal sample, make it gray. So it's telling us one cluster is all tumors and the other cluster is all normals. And that's really good. In this example, we have chosen a set of genes that perfectly separates our samples. These genes, they are very extremely differentially expressed. And when we plot the heat map with this subset of genes, it perfectly separates our samples. If we were to use a p-value cutoff instead of an adjusted p-value cutoff, if we were to include genes that were not differentially expressed, maybe we included all of the genes we tested and did a heat map, it probably would not perfectly separate out the samples. We might have a few tumor samples sprinkled in our normal and a few normal samples sprinkled in our tumor. But selecting genes that are known to split off our samples clearly split off our samples. So this is a good selection of genes here for this heat map. We select our genes and we ask, how well do they separate out our groups? Okay, so I told you that when you're doing your differential expression analysis, clearly define your group levels. So set your tumor samples to be the first group and set your normal samples to be the second group, so that a positive fold change will mean that it's higher in your tumor, and a negative fold change will mean that it's higher in your normal. I know people will use upregulated and downregulated a lot, but those words are meaningless unless you know which group is the positive group and which group is the negative group. So just as a check at the very end, I made these super simple box plots. This was in this particular run through, TICRR was my gene with the highest positive fold change. So I plotted this gene separated by sample group, and I see a positive fold change means that it's higher in my tumor sample compared to my normal sample. And then on the other end, I found the gene with the largest negative fold change, and it was MyoC. And this gene had a higher expression in my normal sample and a lower expression in my tumor sample. And so this tells me I set up my groups correctly. The tumor is the positive side, the normal is the negative side. Okay, so now that we've formatted our data, we've put it in our differential expression analysis, we've looked at the plots that allow us to understand how well did this differential expression analysis go. And now we want to know, do these genes that seem to be differentially expressed, can they tell us anything about biology? And you will see researchers refer to this as pathway analysis or functional enrichment analysis. But there's two types of functional enrichment analysis that we do. The first is overrepresentation analysis. We ask, are there known biological functions or processes that are overrepresented or enriched in our filtered gene list, in our list of differentially expressed genes? So what we do is we get this contingency table. We consider our genes that we've tested. We count how many were not differentially expressed and how many were differentially expressed. Using whatever cutoffs we choose to use, we need to divide our genes into differentially expressed and not differentially expressed. And then we have a group of gene terms, or categories. So for one particular gene term, we ask, are these genes that are differentially expressed, are they in my category? And how many were not in the category? And the same thing for the genes from our data set that were not differentially expressed. How many of these not differentially expressed genes were in the category? And how many of the not differentially expressed genes were not in the category? And with this contingency table, we calculate you could do a chi-squared test or a Fisher exact test. Fisher exact is what we use when some of these values could be small. And that's sure to happen. So it's better to use a Fisher exact test in this case. And we ask, is what we're seeing, does this seem to suggest an enrichment of this category in our differentially expressed genes? This example, I calculated it from one of those web services with a calculator for Fisher exact test, and I found the p-value to be 0.17. And so whatever this category is not enriched in our differentially expressed gene list. Now, in this case, we tested one category. In real life, you might have a whole lot of categories. You might have, I don't know, but we'll talk about the types of categories later. But there might be 200 categories you're testing. So if we just went by p-value, you'd be getting a lot of false positives. So this is another case where we need to do multiple testing correction because we are testing every one of these categories. So what could these categories be? The most popular one are these gene ontology. It defines concepts and classes used to describe gene function, and it tells you relationship between those concepts. There's 3 different types: molecular function, cellular component, and biological process. And the thing you have to remember about gene ontology terms is they have levels. So we start out with very specific terms, and they feed into broader terms, and then you just have biological process, including all of these other terms. So there's levels of broadness and narrowness to gene ontology, and the more narrow term will also be included in the broad term. So there's redundancy in these terms. The other types of functional groups are what we call pathways. So KEGG is one group of these defined pathways. There's also wiki pathways. There's disease ontology. A lot of these pathways and terms and functional groups, they're all described on the Molecular Signatures Database, or MSIG-DB. And if you want to find all of the genes related to one particular function or process, you could go there and search. Another option for functional enrichment analysis is the Ingenuity Pathway Analysis. And you need to sign up for an account through OSU. I think there's like a $300 charge per year for a user for that. And so talk to your PI if you really want to learn how to use IPA. The benefit to IPA is that it can assign a direction to your pathway. It can say, this pathway with your set of differentially expressed genes, knowing the fold change up and down, this pathway is overall up in your data set, or this pathway is down. It can make that up and down designation where typical overrepresentation analysis cannot. Okay. There are also just some really easy copy and paste on a website applications, I use Top Gene more than any of these. It has a really nice interface. You have your list of genes, you paste it in, you get some results. I will go here real quick, just to show one thing that you need to be aware of if you're doing some kind of functional enrichment analysis. So you would paste your gene set here, and then it also has an option for a background set. So if we think about our contingency table here, we need to know how many genes were not differentially expressed. And so in RNA sequencing, we just assume that we're measuring all the genes and we know what is that background of genes that are not differentially expressed. So we assume that everything else has been measured and everything else is not differentially expressed. Maybe in your case, you are measuring a very specialized cancer panel that has 1,000 genes that you know are likely to be involved in cancer. You measure the expression of that panel, you do your differential expression analysis, and then you're doing functional enrichment analysis, and you paste that your list of 25 genes that are differentially expressed as your enrichment gene set. Since you had a targeted expression measure, then you need to define the background set. Your 100 genes that you measured, or I said 1,000, I don't know, whatever genes you measured, if it's something less than the entirety of genes that exist in the genome, you need to tell your functional enrichment program, this is my background. So it knows how to calculate the genes that are not differentially expressed. That's something a lot of people just skip over when they're doing functional enrichment analysis. They don't worry about the background, but it's actually something kind of important. I know in the functional enrichment program I use in Cluster Profiler, they call it a universe. So consider what is your universe and whether you need to correct for that or not. Okay, this is getting long. Okay, so the other way of doing functional enrichment analysis is what is called gene set enrichment analysis, or GSEA. And the difference between this type of functional enrichment analysis is that all your genes should be used. You are not going to filter the genes in your data set for just the differentially expressed genes. You're using all of them. So if you're doing gene set enrichment analysis and you filtered the gene list, you're doing something wrong. So gene set enrichment uses all of the genes. And what it does is for every term that you want to test for, it's going to take the genes in that, no, it's going to take all of your genes. So this is all of the genes in your data set. And then it's going to overlay some term to the genes in your data set. And the genes in that term are these that are marked with the long black lines. So term X, we are marking the genes that are in term X in our entire data set. And so then it's going to go through one gene at a time, starting at the beginning. And it's ranked to these genes. Our entire data set of genes, it's ranked based on fold change or p-value, some sort of ranking. And it's going to walk through saying, start at the beginning for these are just for one side. So for genes that are most differentially expressed for our normal samples, it's going to walk through, and for every gene in the term that it hits, it's going to walk up this score. It's going to add to the score for every gene that exists in the term. If it hits a gene that's not in our term, it's going to penalize the score a little bit. And it's going to keep walking until, let's see, until the score gets penalized too much. And then it's going to start walking down. So it's measuring the score, it's adding if there's a gene in the term, and then it's subtracting if the gene's not in a term. And it's going to just keep subtracting because there's a big gap here. And then there's one gene here, and it's going to bump up a little bit, but it's not going to be enough to bump up the enrichment score any. There's some over here that bump it up where we can see, but still, it's keep going down because there's not as many genes in the term in this end of our data set. And so it's going, once it draws this line, it's going to ask, where was that maximum enrichment score? And that value is our enrichment score. The leading edge subset is going to be this group of genes that exists in our data set and in the gene term that will be listed in the results of this gene set enrichment. And so it's going to do this to all of the gene sets that you want to test. Yeah, and this describes that enrichment score in more detail, but it walks down your ranked list of genes, increasing a running sum statistic when a gene is in the set, and it's decreasing if the gene is not in the set. And then once every gene set gets an enrichment score, those scores are normalized based on the size of the set. And that's what you can use, your normalized enrichment score, to compare set versus set. There's also going to be a p-value and an adjusted p-value. So think about those you can filter on. When I do functional enrichment analysis, I use Cluster Profiler. It's a package in R. It can do over-enrichment, and it can also do gene set enrichment analysis. I do over-enrichment just all by default. That's what I do. If someone asks for GSEA, I'll do that too. But my preferred is over-enrichment analysis. So we define our gene list, possibly a universe list. We tell it the organism that we're using. We tell it the gene set terms we want to use. We're using Go terms here. We're telling it do molecular function. We tell it how to adjust the p-values, what sort of cutoff to use, and what is our gene name and its symbol in this case. Now, I told you that Go terms, they are redundant because one term can be completely within another term. And so you do your enrichment analysis. If you want to remove some of that redundancy, there's a simplify function, and that will compare all of the terms and take out some that might be too similar to your results that you already have. And then another thing that I think is really useful is, let's say you have 3 groups in your RNA-seq data. You compare group A to group B, you compare group B to group C, and you compare group A to group C. So you're doing all of these different differential expression analyses. You might want to know, what are the Go terms that are similar or different between these different gene lists that we get out of our 3 different comparisons. You can do that with the compare cluster function. Okay. And we do that with the formula set up where we have, in one table, we have all of our gene symbols and we have our groups. So we're relating this group comparison to the gene symbols in this compare cluster function. This is what the output to our enrich go function here. This is what the output would look like. So it tells us all of these molecular function terms. It tells us the gene ratio. So how many differentially expressed genes did we find belonging to the term compared to all of our differentially expressed genes? And then how many not differentially expressed genes belong to that term compared to all of our not differentially expressed genes, or all genes? This bottom part is all genes. Okay, it calculates that. This is a Q value, so it's an adjusted p-value. And then it tells us all of the genes from our list that exist in this term. So this is a text file this is a table that I export to a text file for every functional enrichment analysis that I do. So you could go back and find the set of genes related to gap junction channel activity that are differentially expressed in our data set. Maybe you could make a heat map of these 6 genes. If you're going to do KEGG enrichment, you must convert your ensemble IDs or gene symbols to entree IDs. And this can be done with bitter. It's a function or it's a package in R. Yeah. Okay, these are some plot results you can get out of your functional enrichment analysis. The bar plot is, let's see, it's covering up. Floating meeting. Okay, so this bar plot is the length of the bar is based on how many genes from our list belong to this term. The color of the bar is the adjusted p-value. So the redder the color, the smaller the adjusted p-value. There are also dot plots where the size of the dot is related to the number of genes, and the color of the dot is related to the adjusted p-value. This is that compare cluster example that I was showing where we ask, are the genes with a negative fold change are downregulated, and the genes with a positive fold change are upregulated? And what gene terms are enriched in these two groups of genes? So downregulated genes are more related to heparin binding, peptide hormone binding, all of these binding things. They're enriched in our downregulated genes. The other side are upregulated genes. They're related to tubulin. They're related to DNA activity and binding. So they're over here. So there's different functions represented by our positively or negatively changing genes. The cluster profiler can also make these gene concept networks so that they can show structural constitute of cytoskeleton is related to cell-cell adhesion mediator activity based on one gene. These are both terms that are enriched in our data set. So there are a few different ways of visualizing the functional enrichment results that we get out of cluster profiling. So what have we seen here? We have learned some of the conceptual information of differential expression analysis, visualization, and functional enrichment analysis. Next, we are going to look at the script and run through the script line by line and talk about how to set up these functions, these commands. As you do this, think about what would you do next. If this was your personal research project, you did a differential expression analysis, you got a list of genes, you find functional enrichment results, how would you link that back and guide your future research into lung cancer? So think about that as we talk about this as you do your homework later. Okay, thank you.